So we're going to start by talking about the basal ganglia. So the basal ganglia is one of those three motor control regions that we talked about, which included the primary motor cortex and the cerebellum. The basal ganglia itself consists of several regions, and we are, for these purposes, interested in the substantia nigra and the striatum, because that's a dopamine pathway that's involved in Parkinson's disease. We described the basal ganglia earlier as the doorman. So it chooses what signals to go through so that we get proper and fluid movements. You might be getting signals to move one way. You have competing signals for balance. You have competing signals for walking, as well as purposeful movement. And something has to come out as nice, smooth instructions. And that's where our basal ganglia comes in. So obviously, the motor pathways here are more complicated than this, but this will work for us right here. So we start out with dopaminergic cells with cell bodies in the substantia nigra. Those send axons to the striatum, where they're going to release dopamine. Now, we're going to call these substantial nigral neurons because we always base the location of the neuron based on where the cell body is. So these are nigral neurons releasing in the striatum. And the dopamine release here heavily modulates movement control. The problem in Parkinson's disease is that these dopaminergic cell bodies die off. And so therefore the connections and the synapses in the striatum die off. And these slowly die, resulting in a decrease in dopamine release in the striatum. Looking inside the basal ganglia itself, and again, this is a simplified version, what we want to do is we want to balance movement. And we already talked about these dopaminergic cells that start in the substantia nigra and send their axons to the striatum. The important thing that dopamine is doing here is it's affecting the signaling coming out of the striatum, and that's going to go to the actual muscles and control motor movement itself. So dopamine is really only important because of what it does to GABA. Remembering that it's an inhibitory neurotransmitter is actually really helpful here for us because we can kind of simplify it and think of GABA as inhibiting movement. So if you have high GABA being released, you're going to inhibit movement. And if you have very little GABA released, you're going to have a lot of movement. Now, we obviously don't want to inhibit and we don't want to let all movements through. So what we need is just enough GABA release that'll allow us to control what movements go, but have fluid and controlled and useful movement. So if this is the big deal, is how much GABA gets let out, then what we want to see in the striatum is what's going to modify that GABA release. So we have three different types of neurons here that are going to synapse onto that GABAergic cell, and they're going to tell that GABAergic cell how much it should fire. So those dopamine-releasing neurons from the substantia nigra are going to inhibit the firing of the GABA cell. We have acetylcholinergic cells that live in the striatum that are going to excite the GABAergic cell. And we have glutamatergic neurons that are coming from the cortex to the striatum that are going to also excite GABAergic cells. Okay, so let's look more closely at those synapses within the striatum itself. So here's the striatum again, and we've got the dopaminergic neurons coming in, releasing dopamine, and what they're releasing dopamine on is this GABAergic cell. Dopamine is being released onto D2 receptors. And if you think about, we talk about neurotransmitters like glutamate as being excitatory neurotransmitters or GABA as being inhibitory neurotransmitters. But really it's not the neurotransmitter itself chemically that's excitatory or inhibitory. It's the effect of the receptor. And so glutamate's only the excitatory neurotransmitter because all glutamate receptors are excitatory. And the same thing with GABA, all GABA receptors are inhibitory. So you've got other neurotransmitters that have either excitatory or inhibitory receptors, and you have to know which one is being activated to really understand what's happening in the circuit. So here, dopamine is being released onto, in this circuit, D2 receptors. And as I said, D2 receptors are inhibitory type, so those are going to inhibit the activity of the GABA cell. So that's inhibiting it. We also have those cholinergic neurons. That's releasing acetylcholine onto, in this case, a muscarinic subtype that is excitatory. That's our positive here. So that input is going to be excitatory to the GABA cell. So now we have this excitatory input. Glutamate, similarly, coming in, and all glutamate receptors are excitatory. This case, it's the NMDA receptor. And so again, that is exciting the GABA cell. So now, when the GABA cell is trying to decide what to do, whether to fire an action potential, how fast to fire them, what it's looking at is going to be the sum of all these inhibitory and excitatory inputs. So what we want is we want the excitation by the glutamate to balance out the inhibition by the dopamine to the point where we get the proper amount of excitation, proper amount of GABA activity. And if you get the right amount of GABA, you get good movement control. It's fluid, you don't have tics, and you don't have tremors. If you don't have enough dopamine coming in, or if you have too much excitation, then you're going to increase GABA release too much. And if you have increase in GABA release too much, then you're going to decrease movement and you're going to cause tremor. This is just a more graphic version for those of you who are more visual. In the middle here is what we want. We want controlled movement. So we want that dopamine inhibition to balance out well with the glutamate and cholinergic excitation. In Parkinson's disease, what we have is that decrease in dopamine, and we don't actually change our glutamate or acetylcholine levels. So these are actually still the same. But the relative difference means that there's more acetylcholine and glutamate relative to dopamine. So what ends up happening is you get too much GABA release, and that's when you get those slowed and blocked movements, and you get tremor. And here is a video link for explanation of tremor if you're interested in it, but it is really complicated, and unless you're really interested in it, don't worry about it. So down here we saw Parkinson's disease, but what happens up here if there's too much dopamine can occur often as a side effect of some dopaminergic agents. So what you can see here is if you increase the dopamine or you increase the ratio of dopamine, then you are going to inhibit those GABAergic cells. So increased dopamine inhibits the GABA cells, while glutamate and acetylcholine are going to excite them. So now you've got too much inhibition, and you don't have enough GABA release, and now you have uncontrolled movements. So you're not inhibiting movements anymore. And we're going to see that we have specific movement disorders that occur as a result of too much GABA. So we're going to see that we have movement disorders that are side effects as a result of too much dopamine. Okay, so at the risk of being repetitive, we're going to look really quickly at what dopamine cell death does to GABA release. So here we have those substantia nigra dopaminergic neurons. They're dying. No more dopamine onto the GABA cells. So now all you're left with is excitation. GABAergic cell becomes extra excited. GABA release goes up. And since GABA is inhibiting movements, with that increase in release, we get decreased movement and we get tremor. So that's exactly what we're seeing when we're looking at the movement disorders in Parkinson's. Now, we talked about how Parkinson's, we mainly think of the movement disorders, but we also have some cognitive and psychiatric issues. The way to think about that is this may be the pathway that is most affected, this dopamine pathway in the motor system, but there's also dopaminergic cells doing other things in the brain, and some of them might be involved in some psychiatric things like reward and addiction and mood, and some of them are probably also involved in cognitive processes. So when you start messing with dopaminergic neurons, we might be seeing these motor issues in Parkinson's as our predominant symptoms, but that dopaminergic influence is also getting altered in those other regions.